Every city runs on supply chains. Factories send steel to warehouses. Warehouses ship goods to markets. Residential areas send workers to factories. These supply routes connect different districts into a functioning economy. In tissue, cells communicate through similar molecular supply chains, ligand receptor signaling pathways. Think of each cell type as a district in our city. The tumor core is the industrial zone. Immune cells form the security district. Terminal cells are the infrastructure department. Each district has its own products to export and needs to import from others. CellChat maps these trade relationships at the molecular level. A supply route in our city connects a sender to a receiver through a specific product. In molecular terms, a ligand produced by one cell type travels to a receptor on another cell type. CellChat scores each potential route by how strongly the ligand is expressed in the sender and how strongly the receptor is expressed in the receiver. Let us zoom into one supply route. A sender cell produces a ligand molecule like a factory shipping a product. The ligand travels through the tissue microenvironment. When it reaches a receiver cell, it docks at a specific receptor like goods arriving at a warehouse loading dock. This molecular handshake triggers downstream signaling. CellChat assembles all these individual routes into a complete communication network. Each cell type is a node. Each significant ligand-receptor interaction is an edge. The network reveals the full architecture of cell-cell communication, which districts talk to which, through which channels, and how strongly. From raw sequencing reads to a complete communication network, this is the spatial biology pipeline. Next flow processes the raw data. Banksy identifies spatial neighborhoods. Spatial finds the genes that define those neighborhoods. And CellChat reveals how those neighborhoods communicate. Together, they transform a tissue section into a living, connected city map.